Adaptive Immunity breakout session. Me and my amazing co-chair, Dr. Petros, welcomes you. We have 3 amazing talks lined up for you. First speaker will be Amir Horowitz from Icahn Medical School at Mount Sinai. And next up will be Dr. Madhuri Koti from Queen's University School of Medicine. Dr. Swatik unfortunately couldn't make it today because of a family emergency. He had to fly out last night. And then lastly, we'll end with Dr. Chan. So this session, I really want the questions to all come in. I want it to be an engaging session. Just bring all your ideas and we can have a discussion. But before we get to the talks, I wanted to give a very brief introduction to the topic. So immunity classically is divided into innate and adaptive immunity. So innate immunity is considered like the first line of defense, and it consists of cellular and biochemical barriers that are in place even before infection or tumor develops, so that it can respond very rapidly to the stress, whether it is infections or tumors. It consists of phagocytic cells like macrophages and neutrophils, cytotoxic cells such as NK cells. We'll hear talks on it today. And then dendritic cells that also play a very critical role in antigen presentation to T cells. It further includes mast cells, and the role of mast cells in bladder cancer specifically is not really known. It has a dual role, both in pro-tumorigenic as well as tumor protective roles. And then the group of proteins known as complement, which are present as inactive forms but can activate a cascade of downstream signaling pathways leading to lysis of stress cells. Then adaptive immunity, so the top 2 features of the adaptive immunity is its specificity as well as a memory response, which includes a heightened response after repeated exposures of the same antigen. And the 2 most heavily researched members of this group are the B cells. I think we'll hear about it from Dr. Koti today, and then T cells, including the cytotoxic CD8 positive T cells and the different subsets of CD4 T cells. However, this idea that the innate immunity is this quick, non-specific arm of immunity is not really accurate at this moment. Over the last decade, we have seen that NK cells are actually very tightly regulated by a group of activation and inhibitory receptors, and it's actually quite an intricate network. Also, in recent years, we have seen the identification of almost memory-like NK cells that show heightened production of cytokines after repeated exposures of the same antigen. So there is a lot of crosstalk between the 2 arms of immunity, and as we are slowly developing our ideas of the innate immune cells, they almost play an equal important role as the adaptive immunity. So this shows that how the different groups of innate immune cells developed almost synergistically with the subsets of T cells. You can see the CD8 positive T cells equating with the cytotoxic NK cells, and then ILC1s equating with the Th1 response regulated by T-bet and secrete interferon gamma. Then the ILC2, which is the Th2 innate counterparts regulated by GAT3 and secretes IL4, 5, and 13 upon activation. And lastly, the Th17 like ILC3 cells that is regulated by ROR gamma T and secretes IL22 and 17 upon activation. All these cells help each other in producing similar kinds of immune response, but the importance of the innate counterparts, especially in bladder cancer, is not completely understood. So what we know so far about this cells in bladder cancer. So first, the most popular pathway is, of course, the innate checkpoint, the immune checkpoint pathways like PD-L1 and PD-1 and the CTLA-4 axis. So both of these axes has been targeted by the checkpoint inhibitors, which are approved for late-stage and metastatic bladder cancer, that helps in the activation of T cells. But interestingly, if you look, the markers that, like PD-1, is also expressed on innate cells, like dendritic cells, as well as NK cells. NK cells also express the same immune exhaustion markers that has been traditionally seen on T cells. So when we are using the immune checkpoint blockers, it's not only affecting T cells, it also may have an important role on the function of NK cells. Our group have shown that there is significant infiltration of NK cells in bladder tumors, but those NK cells are dysfunctional, has lower cytotoxicity, low expression of CD56, and low cytokine production as well. NK cells seem to have a more dysfunctional phenotype in bladder cancer, and we have shown that the CD56 dim cells accumulates in higher stage of bladder cancer, whereas the CD56 bright NK cells correlate with improved survival in patients with high stage bladder cancer. We also saw infiltration of this Th17-like trans-differentiated ILC1s in bladder tumors, and so that's also an axis that is worth investigating in bladder cancer. Another recent study also saw the important group of ILC2 that helps in the recruitment of myeloid-derived suppressor cells. And the ratio of this ILC2 recruited MDSCs and CD8 positive T cells has been shown to modulate the BCG response. Another interesting study that came out also saw this unique population of double positive CD4 and CD8 T cells that seem to have a more of a Th2-like phenotype and equates with increased tumor growth. But basically, what I want you to get from this figure, that as you can see, that there is a lot of different ways that the innate cells and the adaptive immune cells share the same axis and are talking to each other. So kind of there is a lot of overlap between these two arms of immunity. I don't need to go over the approved immune therapies for bladder cancer to this group, but the reason I put this slide here is that we can see that there are different kinds of immune therapies that are approved for bladder cancer. But when we talk about them, mostly we think that when PD-1 or PD-L1 immune checkpoint blockers, they are activating the T cells. But as I wanted to point out that this immune checkpoint blockers equally has a very important role in the innate immune axis. And maybe it's time to think about how we can look at the innate immune axis and improve the response or identify novel agents that can increase the efficacy of the already approved therapies that are in the clinic. So even though the session is named as innate versus adaptive immunity, this is not a debate of which one is more important. We wanted to focus on the synergistic application of both arms of immunity for development of novel therapies and markers. And a perfect example is actually the pre-807 trial that is led by the amazing Dr. Grivas here. And it's a very interesting combination that combines the anti-PD-1 with anti-KIR antibody in a neoadjuvant setting. So the trial schema is listed here. You have the patients undergoing TERT. Then there are two cohorts that receive Nivo 4 weeks apart in 2 doses, and then cohort 2 that receives the Nivo and anti-KIR. So anti-KIR is a KIR2D pan-blocking antibody that helps in the activation of NK cells. KIR is an inhibitory receptor on NK cells, so blocking the anti-KIR helps to activate the NK cells. So this one was presented in ASCO 2021, but now Dr. Grivas has some new data, and the manuscript will be coming out next week. So if you want to discuss this trial with him, he'll be more than happy to talk about it later. So this is the trial that sets the perfect example of how we can use the both arms of immunity and can lead to novel combinations of immune therapeutic strategies. And with that, I will hand over to the speakers, and we're excited to listen to their talks.
All right, can everybody hear me okay? Great. So it's the big green one, is that?
Green goes forward.
So I pressed it once, I'm trusting.
Yeah. I think they're loading the slides.
All right, well, here we go. So hi everyone, my name is Amir Horowitz. I'm an associate professor of immunology and immunotherapy and of oncological sciences at Mount Sinai. Today I'm going to be talking about relatively new to this community, inhibitory axis that's playing a fairly dominant role in driving resistance to BCG and designing some strategies for how we can exploit that immunotherapeutically. So I just want to start by, well, these are my disclosures. I just want to start by taking the time to truly express my gratitude to BCAN, our reviewers, and our clinical partners. Very grateful for having received this first ever clinical trial award. And I thought I would just start off by taking the time to just briefly describe the trial and then kind of go back and show you how we got here. And so what you're looking at here, is the ENHANCE trial. It's sort of spelled out here for you. It's a trial targeting NK cells and CD8 T cells, with the idea being, can we dually target these cells, appreciating that they play highly redundant roles, but at different temporal windows of immune activity. And so can we use one to boost the other? This is based on a combination of using an NKG2A inhibitor called monalizumab alongside durvalumab, starting off with BCG unresponsive, non-muscle invasive patients with high-grade disease, where it's a single-arm trial broken down into two cohorts. Cohort A is CIS only, where we have a primary endpoint of safety and CR at 6 months. Cohort B, which is really more reflective of the biology that I'll be showing you all in the subsequent slides, is actually papillary plus or minus CIS. We'll be asking for secondary endpoints, overall survival, event-free survival at 12 months, cystectomy-free survival at 12 months as well. And then as an exploratory aim, for which I'm very excited about, our preliminary data suggests that HLA-E expression on the primary tumor has a predictive role in the strength of response to blocking NKG2A. So it sounds probably complicated, I'll just show you the data. Okay, so aim 1 of this study is to conduct the trial, and then aim 2 is ultimately to tease this out through multiple modalities such as multiplex IHC, spatial sequencing, high-resolution imaging using mass cytometry, as well as cytoph on urine and blood. Okay, so how did we get there? I'm going to walk you through some high-level findings based on standard of care BCG that the work very much in partnership with John Sfakianos, who's in the audience here. And so I don't need to go through the treatment regimen for this audience, but just consider that pre-treatment we get tumor blood and urine. At time of each cystoscopy, if tumor, we get tumor, and we capture blood and urine throughout this process. At a high level, I certainly don't have to describe steps 1, 2, and 3 to you all, but we, as well as many others, have described a very strong local Th1 response to BCG, to the actual treatment. This is not particularly novel. We've used some very high resolution techniques to really tease out the key players, and it seems like the local response is heavily mediated by myeloid cells such as DCs and macrophages, effector memory CD4 T cells, which essentially is providing a pool of type 1 cytokines that are extremely effective at activating NK cells, CD8 T cells, both locally and peripherally. What we're finding is that with so many repeated doses of BCG, we're finding that every single patient is actually responding to BCG as a response to the treatment in the form of inflammation. I'll show you some of the data, but essentially we profiled urine, we profiled plasma using a platform called the Olink proteomics, where you can profile 92 soluble analytes simultaneously. And we looked every week on therapy, compared it to before therapy. We found that the responses, they never drop officially to the baseline pre-treatment levels, and they just keep going up and up and up. And so this chronic activation, it has a net result that it's driving expression of inhibitory checkpoints on NK cells and T cells. And then very importantly, tumors, if they recur when they recur, they're met by a functional status of our immune system. And what we're finding, that I'll show you in the subsequent slides, is that a subset of these tumor cells, and this is, we're observing in every single patient, a subset of the tumor cells in the unresponsive time point have extremely high levels of HLA-E, and to a lesser extent, but certainly significant, PD-L1 as well. They even have like the don't eat me signals and other things popping up. Okay, so just building on sort of what Neelam was telling us, just with a little bit more resolution for you, NK cells act as a population. They are not antigen specific in the true sense of a T cell or a B cell. Their fate, if you will, for remaining inhibited or becoming activated is the net sum in signaling strength across a variety of activating and inhibitory signals. These activating signals all have short cytoplasmic tails lacking tyrosine motifs. They associate non-covalently with adapter proteins, whereas the inhibitory receptors, they all encode directly inhibitory tyrosine motifs, and as such, inhibition typically outweighs activation. It's probably a good thing, because as these cells develop predominantly in the bone marrow, if they didn't have these brakes already on, we would probably all have quite severe autoimmune-like diseases. So this is a way that they self-regulate themselves. We published the first study of human NK cells by Saitov back in 2013. Before that time, these cells were thought to be a relatively homogeneous population, and in fact, it's really not the case. The more markers you add, the more variability you see. And so the main functions of these cells is that of surveillance. They're looking to see what does healthy HLA class 1 look like, which predominantly for these cells serves an inhibitory signal to them. But as many of us know, one of the earliest alarm bells, immune events to happen after transformation, infection, or even like chronic immune stress is that you get modulation of HLA class 1. So the better that these cells are trained, the better honed in they are to sense these early immune events, where these immune events are thought, the dogma, to be a strategy by the tumor to evade a T cell response. And I'm very excited, I'm going to dive into some very novel findings that we published recently and sort of dispel some of these myths. So we all know the landscape of checkpoint blockade therapies. This is just a list summarizing some of the major targets, but considering that these targets, while we appreciate them mostly around T cell biology, they really are very dynamically expressed as well by NK cells. And so what Neelam was discussing before in terms of the KIR biology, these are receptors. It stands for killer cell immunoglobulin-like receptors. There's 14 unique genes, and they all have their own alleles, and collectively, an individual can have anywhere from 4 to 20 of these genes. So there's tons of diversity on the human population level, and it's in fact the second most polymorphic gene family, second only to HLA, and it's encoding proteins that interact with HLA. This is a system, mind you, that's been evolving since before RAG genes. So just consider that this is our ancestral immune system. Today, however, I'm going to be talking about this other pathway, CD94 and NKG2A. For the purposes of this talk, I'll just refer to it as NKG2A. Okay, so emerging data has been showing that NKG2A can actually be induced on CD8 T cells as well. The idea here is that CD8 T cells, we all know them to express PD1, CTLA4, et cetera. NKG2A, I'll show you data in a moment. It comes up after all these other checkpoints, and it serves a function where these cells appear to be diverging from classical exhaustion to take on evolutionarily ancestral functions that allow them TCR independent manners of activation, and it seems to be in response to a microenvironment that has lost HLA class 1 on target cells. All right, so we published this paper late last year in Cancer Cell, focused on sort of all comers of bladder cancer, but the majority of it was really capturing late-stage muscle-invasive disease. And we found both in TCGA as well as in 3 different cohorts with Genentech that the abundance of NKG2A on CD8 T cells, not on NK cells, is actually pretty strongly predictive of response, both in terms of prognosis survivorship as well as response to treatment. We found that NKG2A is specifically upregulated after PD1 and after almost all the other checkpoints that we looked at. And we found that these cells can react to HLA-ABC deficient tumors, as well as HLA-E deficient tumors. But in the presence of HLA-E, these cells are highly inhibited. So we also found that NKG2A blockade allows us to tap into that biology. So this is just a snippet of our data. What we did was we took CD8 T cells directly from the tumors that we received from John in the OR, and we ran mass cytometry or cytoff. And what we found, just doing some basic unbiased clustering of the data, where every row here is a unique phenotype found in the data set. And so what we did was we actually organized these data according to the abundance of NKG2A in each of these clusters. And what we did was we simply co-cultured these cells for 6 hours in the presence of a tumor line called K562 cells that endogenously lack any HLA class 1. They lack HLA-E as well. So we have a system that we can transduce HLA-E to be expressed at high levels, and thereby we have like a nice model system to really see, do these cells require HLA-ABC presented antigens? And what we found perhaps most striking was this very strong anti-correlation between NKG2A on these cells and CD28 on these cells. So appreciating that we're not all immunologists, when we think about a tumor-specific response, you're talking about 3 critical steps that are considered to all be fundamental. First one is the TCR recognition of the MHC peptide complex. Signal 2 is CD28 engaging CD80, and then signal 3 are cytokines. So if you have no CD28, this is like, you know, in terms of like popular science, what's out there today, that's the definition of classical exhaustion, is that you're not going to get that response. And in fact, when you look across these 7 different cytokines, chemokines, what you find is that they organize naturally, that the more NKG2A these cells have, the less CD28 these cells have, the more abundant this TCR-independent response is. We can even pull these markers out separate. FSI just stands for Functional Stimulation Index score, but we can even show it on the individual level. Okay, so we wanted to see sort of what is actually driving NKG2A acquisition by these cells. It seems if they're really diverging functionally, it would be really nice to know what is actually pushing this forward. And so it just so happened that there was a study maybe a decade ago that was playing around with TGF-beta and IL-15, and they found on the surface of T cells NKG2A can come up. So what we did here was we went back to the tumors that we got from John, And we essentially isolated total CD8 T cells, and we sorted them such that we took only the double negative population, meaning negative for PD1, negative for NKG2A, and then we cultured the cells in the presence of TGF-beta and IL-15, or just IL-15 alone. And what you can see on these representative plots here is just the difference in induction of NKG2A when you actually have TGF-beta present. Shown just as a summary, it's about 25% or more of this population in that reaction are inducing NKG2A. And when you consider now co-culturing these cells either with the wild type tumors that lack HLA-ABC, but they also lack HLA-E, as compared to the same tumor line, but now where it's been transduced to express very high levels of HLA-E. We see this fundamental difference in the activation response. Now, very importantly, what we found is that when you look at HLA-E expression on the primary tumor from which these cells derived, that's what you're looking at on the y-axis here, as compared to the strength of response to monalizumab in vitro. So it's very curious, and it certainly needs to be validated, but it suggests that there's almost a memory component built into this recognition and response, similar to what we've profiled around NK cells, which we actually call that process education. So showing you here is single-cell RNA sequencing data from the same tumor-derived CD8 T cells. And what we did was what we call a trajectory analysis using a software package called RNA Velocity. Essentially, for any 2 or more genes, you can profile the relative initiation of transcription, and you can do it in such a manner that you're doing this sort of multi-pronged approach. And so every line here is a cell, and we broke this down into 5 different clusters. And so what you can see at the top here, cluster 1, the idea here is that the genes coming up in cluster 1 come up as a model before the genes coming up in cluster 4 down here. And so we see this very large collection of checkpoint inhibitors coming up early. And the gene for NKG2A, KLRC1, is here. There's a number of genes as well, SH2, DBA, and XBP1. These are genes associated actually in silencing TCR signaling. So it seems that there's a lot of compensatory things happening here that are all falling in place supporting this observation that these cells are actually diverging from classical exhaustion, and it might actually reflect a third wave of adaptive immunity, which I'll get to at the end. So just really briefly, so I'm not running out of time, this is in settings of BCG treatment here. What you're looking at are representative multiplex IHC plots from one patient at the naive pretreatment time point or at the unresponsive time point. What we found, and I think others are seeing this as well, is that there's a relative void of immune infiltration in untreated non-muscle invasive bladder tumors. I'm sure there's examples that, there are caveats deviating from that, i.e. like UTIs or other things, but overall, we see really very little immune traffic in the bladder of untreated individuals. And what we found was that every single tumor in the resistant time point had a lot of immune infiltration, and they were entirely trafficking around tumor nests that had higher expression of HLA-E, and they were separated by tumor nests that didn't have HLA-E by distances of anywhere from hundreds to thousands of microns, very small space, and you're seeing all the immune traffic going to this one type of tumor and not the other. It was very curious to us. Something about that had to be instructing this. And so based on the fact that we ran these early analyses and found that interferon gamma from NK cells and T cells is really seeming to strongly correlate with disease resistance, it's a little counterintuitive, but it suggests the process of adaptive resistance by these tumors. We summarize it here on the right. As I mentioned, PD-L1 certainly statistically increased. It's less sensitive of a marker, it seems, than HLA-E, shown on the bottom. We ran some proximity analyses asking the question, based on the observation that NK cells and T cells are clearly recruited to these tumor nests, why are they not killing them? And so we started off by profiling some proximity analyses, and shown here is taken from our BCG naive samples versus the unresponsive samples. The x-axis here is measuring proximity to the tumor from the perspective of the tumor. So anything to the left of the 0 means that that cell is physically within the red boundary on those slides. So it's officially infiltrated. And you can just see a very clear difference here. The y-axis only goes to 80 cells, period, per millimeter square of tissue, as compared to 8,000. So we saw this really big increase. We can profile these further. This is just showing a representative example, but showing that NKG2A biology is very relevant in this microenvironment, both on the CD8 T cells and on the tumors, on the NK cells. All right, so this is, I think, the last data slide. But what we did next was we wanted to really drill down into the signature, this program of the tumor. So what you're looking at here is everything from the lens of the tumor cells. And so we ran what's called spatial sequencing. You take a section of tissue on a slide, and you can sort of overlay it on a special slide that has chips with probes, and it's essentially allowing you to get whole transcriptome amplification at close as possible to single cell. So you can see these individual spots. They represent precisely 55 microns in diameter. And so depending on who you ask, you're looking at 3 to 5 cells, 5 to 10 cells. It's very close now that we're getting to true single cell resolution, but now while maintaining spatial integrity. So we can see what are the genes that are driving these cells. And so as an example here, if you look in panel A, what you're looking at is sort of a continuous expression of HLA-E, just color-coded from light to dark blue. And then we were able to map for you CD8 T cells, NK cells, and regulatory T cells. And what we found is that you see a very strong correlation, even on this platform, very strongly supporting what we found by IHC. So this idea that HLA-E bright tumors might be physically bringing cells to them, you know, it's the chicken or the egg. Is it that the T cells and K cells are trying to find a certain type of tumor cell? Or is it that the tumor cell is sort of self-aware of its microenvironment and it's bringing it over there? But at a high level, there seems to be evolutionary advantage here by the tumor. If you have, crudely speaking, 2 types of tumor cells in this immediate microenvironment, where one has a very strong cytokine activation signature that drives increased expression of inhibitory ligands, and the other doesn't, there seems to be a strategy of sparing these HLA-E negative tumor cells from what otherwise might look like an easy mark for tumor destruction. And so that's what we're seeing here. We profiled expression of chemokines actually coming from the tumor cells. So every spot that we call a tumor cell, we left a lot of data on the table just so that we could be extra rigorous in our analyses. These are spots that have 99+% signatures that are truly unique to tumor biology. So while it's a smaller overall analysis that we performed, it allows us to really bring in that confidence. And we see as an example here, CXCL9, 10, and 11, which are very critical for recruiting cells expressing CXCR3. And there's a strong correlation with HLA-E expression in these chemokines. What you're looking at here, each dot is a tumor spot, and color-coded such that in orange are the BCG-resistant tumors and blue are the naive tumors, where 0 means that you're physically contacting the tumor spot, and 4,000 is like as distal away as possible. And so you can very clearly see here that there's 2 types of tumor phenotypes within the resistant tumor, but they're extreme, they're bookends. And then in the naive space, it's kind of just randomly distributed. But overall, there's just not many immune cells. So I'm going to wrap this up now. This is making us think that there's somehow a third wave of adaptive immunity that people haven't defined. These NKG2A expressing CD8 T cells are 100% memory T cells. You only ever see it in settings of chronic activation and sustained prolonged activation. And these cells are not terminally mature. So they've still got function, it's just not through TCR-dependent mechanisms. So we demonstrate that NKG2A is acquired by these cells during this program. We demonstrate that it allows them to take on TCR-independent innate-like features. And could this actually represent a third wave? This has got nothing to do with bladder cancer. We're seeing this in settings of autoinflammatory diseases, and we're seeing this in other solid tumors as well. So what I hope I convinced you about is that BCG therapy induces a very strong Th1 response, and we're actually seeing this in all patients. Interferon gamma is correlating with adaptive resistance by the recurring tumors. This strongly correlates with activation response to interferon gamma produced by NK cells and CD8 T cells. And these cells are enriched in bladder tumors of patients treated with BCG. So the question is, we know that they're getting there, we know that they're not killing them, and this seems like a very strong dominant inhibitory axis. Does this allow us to exploit this for immunotherapeutic strategies? So this is sort of to bring us here where we got to in terms of getting the BCAN award, which again we're very grateful for. And understanding BCG-mediated immune activation and exhaustion versus the timing of tumor recurrence, I believe, is critical. If you're someone who has very rapid recurrence, chances are your immune system hasn't been chronically activated as long as someone who develops tumor recurrence, say at like week 36 of treatment. And so these are features that with every repeated dose of BCG, you're getting a bump in activation potential, but you're also getting a bump in expression of these inhibitory receptors, suggesting that the timing of tumor recurrence is critical. So the last couple slides are meant to just be provocative in the hopes of stimulating questions. BCG did not start off as a treatment for bladder cancer, let alone for melanoma. It started off as a vaccine to protect against Mycobacterium tuberculosis, a very closely related pathogen. BCG as a vaccine is delivered before exposure to TB, whereas bladder cancer, you're first removing the tumor, you're giving BCG, such that if you ever get tumors recur, maybe the clinical folks here are thinking that there's like a memory response to the BCG. My question for us is, why are we not delivering this in the presence of tumor? This is a non-specific activating stimulus, and it's profoundly activating. So why wouldn't we want to deliver BCG in a setting that brings the T cells and NK cells to where they actually see tumor cells, then resect the tumors, such that if tumors ever come back, you truly have immune memory to the tumors. This is something that we're playing with now. And so there's a number of unrelated pathogens to BCG or TB, where BCG has quite established what we call heterologous immunity. And so the question to us all is, are we really harnessing the benefits of BCG? And on top of this, we're dealing with global shortages of BCG. So I think there's an argument to be said that if we could find a way of using this more effectively, there's like multiple layers here that benefit. And so just to describe for you sort of what this looks like, in a setting of neoadjuvant BCG installation, this could indirectly recruit bystander CD4 and CD8 T cells. Some will be restricted to tumor specificities. And so what we all probably agree on very clearly is that the quality of early tumor control ultimately determines the availability of these tumor epitopes, which will affect the priming of a true tumor-specific response. And so the availability of these tumor epitopes, the idea is that they would improve antigen presentation and priming. So this is the last slide. This is just a schematic of a study that John and I are very much in design of, where we're comparing standard of care therapy, shown up here, to a 3x3 design where one group will get one installation of BCG neoadjuvant, followed by resection, followed by immediately being put onto standard of care. Compare that to 2 installations or 3 installations. And the idea here is that it'll allow us to track development and expansion of both BCG specific and BCG bystander CD4 and CD8 T cells in the blood and the tumor tissue. It'll allow us to profile a therapeutic response to BCG requiring tumor tissue after BCG in non-recurring patients. This is literally what took me and John 4 years to get our R01 on this topic. It was the single most repeated criticism is, why aren't you measuring HLA-E on the tumors during treatment? Like, how you're not following that this is an adjuvant therapy, but the criticism holds in that, the burden of proof is on us. So this would allow us a strategy to engage that. It also provides a platform for testing novel combination strategies, which I think we're all appreciating will become very important. So with that, my partner John, instrumental to this. Additionally, Matt Galsky, Nina Bardwaj at Mount Sinai, a bunch of external collaborators from this community, which I'm extremely grateful to. And I'll stop there. Thank you. Are we taking questions?
We can do it at the end. Alex, let's do it now, real time, so the questions are fresh. Alex, and I see,
Really great presentation. If you think about where BCG is the most effective, it's going to be in CIS, correct? And there, you actually don't have the therapeutic resection, and your model basically is as close as it gets. Now, have you had, and again, CIS is difficult to study because of the amount of tissue, but have you any indication about what's happening with your model, the NK2, in the setting of CIS, or at least a couple of?
It's a fantastic question. We realized this actually as we were designing this clinical trial. This whole time I've been profiling papillary disease and starting to realize from the community, the clinical trial designs, everybody seems focused on CIS and complete response rates at 6 months. Maybe it's wrong to say everyone, but that seems to be a theme. And so really just to fit this within the scope, we set it to CIS. We have not profiled nearly enough, so I want to raise that caveat. In settings of BCG resistance where you're looking at CIS, you see immune traffic, but you're not seeing the same degree of HLA-E expression, perhaps on the tumor cells. It's a very small data set, mind you, so we have to validate this, but just to be clear, when you look in settings when you have papillary and or CIS, and you're looking at these tumor nests, 50% of the tumor cells that they're aggregating as nests, it's very high HLA-E expression. So it's comparable to immune cells.
And very briefly, have you had the chance to see what's going on in terms of trafficking in the granulomas?
I'm not. Yeah, it's a great question though.
Hey, then Vayanowski, Center. Fascinating talk. Thank you so much. I'm always fascinated about, we have so much wealth of data on these immune biology correlates over time, and then when it comes to understanding the best therapeutic space for drug development, it becomes so hard, and we're sort of make those arbitrary decisions. So, we're participating in a late line development, early development of another NKG2A antibody, and I thought your descriptions there of the biology over time and the expression in NKG2A was fascinating. So a lot of the development happens in post-IO patients, and I know you talk about BCG and the biology you showed is about BCG, prior BCG, but if you think about prior PD-L1 in a metastatic patient, do you think the best NKG2A patients are upfront in combination with PD-1 or post-PD-1? And then the post-PD-1 question always comes to, is it the PD-1 refractory patients? Is it the patients who are, and refractory often means progressing within 3 months of prior PD-1 or never responded to PD-1? Or should it be in patients who responded to PD-1 and now are no longer responding? What do you think?
There's a lot there to unpack. I would say we have therapeutic response data around NKG2A, and it's entirely in the muscle invasive and metastatic setting. So it might be a little off topic to what you're talking about in BCG, but there are some important considerations here. One is that in later stage disease, and I didn't really drive this point home, HLA-ABC is all but diminished at the protein level on bladder tumor cells. It's important to say. We ran a cohort where we saw 0 loss of heterozygosity at the DNA level, and yet at the protein level, it's gone. We had immune cells in the same tube, staining beautifully. HLA-E is highly variable. So if you consider a setting where you remove HLA-ABC, and you're accounting for HLA-E being low versus high, it stratifies that response pretty cleanly. In terms of non-muscle invasive disease, there's absolutely nothing special that we've seen pre-treatment about HLA-E. We don't even see PD-L1 that much in the pre-treatment, outside of probably these caveats where you have inflammation due to something else. So in terms of PD-1 in the BCG resistant space, John was running the CREST trial. I don't know if you want to say anything about that, John, but I don't know if we had a good reflection of the overall trial in terms of response or not.
So we have done some spatial sequencing on those. I don't think I can really speak to the data yet about it, but I'm not very impressed. So for your question of how do we combine treatments, which stages should we combine treatments, sequential and so forth. So I think in the BCG naive setting, PD-1 is not going to add much because we just don't see any activation until very further on in the BCG state. So I think you would need to do sort of a year worth of maintenance of checkpoint in the BCG naive, which I'm not sure toxicity profiles and so forth are worth actually doing. For your question in the advanced stage, so Amir, we do have lots of data in the platinum resistant cohorts looking at PD-1, PD-L1, and KGY, et cetera, and I mean, I'll let you speak to that. But yes, there is probably a role for monalizumab or any NKG2A plus or minus PD-1 or PD-L1 in the advanced setting. I think it would probably be more sequential or after PD-1 rather than in combination. But I mean, Amir can speak to that a lot more.
So PD-1 in our hands, when you're looking at the NKG2A negative cells that are CD28 positive, they're very sensitive to PD-1 signaling. When you look at the CD28 negative cells that are NKG2A negative or positive, they start really losing actually sensitivity to those pathways. I think it's just because they're becoming more polarized towards functional energy. So I guess there's a lot more than these NKG2A positive cells in there. I will say though, in cisplatin resistance, and we have a small data set thus far, looks extremely similar biology to BCG resistance. And so we have some single cell RNA-seq data and some CyTOF data showing that you have a really massive increase in NKG2A expressing cells, be it CD8s or NKs. Yeah, but sure.
Amir, fantastic talk. So we know that as tumors progress, from the primary to the metastatic state, there's a loss of HLA, genetic loss, deletions as a mechanism of immune escape. So I wanted to ask you what your insights are. What do you anticipate happens to HLA-E as the tumor progress, and also what do you expect would happen in your BCG, so through the natural history of the tumor from primary to metastatic, but also in your trial as you treat with BCG, what do you think will happen to HLA-E?
So similar to HLA-ABC, HLA-E is very sensitive to inflammatory signals. Very unique to HLA-E, in order for it to reach the cell surface, it requires binding a very specific peptide. And if you have cytomegalovirus infection, that becomes a bit murkier picture, but the 40 to 50% of us that are CMV negative, it requires a peptide specifically derived from HLA-A, B, or C. So this is complicated, but these two systems are fundamentally linked in that the exon 1 of a classical HLA class 1 molecule, that gene, exon 1, it encodes a 20-former amino acid that serves the function of shuttling the polypeptide into the ER, but it's enzymatically cleaved, biodegraded into a highly conserved monomer peptide. So outside of HLA-ABC, the only other HLAs that can do it are HLA-F and HLA-G. And I'm curious, anyone in the audience that looks at RNA sequencing data in Bladder, I would wager that you're seeing high levels of those genes pop up. I mean, we are anyways. But so what we're seeing is ABC can go away. That accounts for the CD8 T cell response, but you have these non-classical HLAs that suddenly are solely sensitive to inflammatory signals, and they provide provision of that leader peptide to HLA-E.
So you think even with the loss of HLA, B, and C, that pathway is, due to compensatory upregulation of these non-classical?
Yeah, and so our studies with Genentech, we have 2 studies. One is in Bladder, and one is in 4 cohorts of their non-small cell lung cancer trials. All Atezo, but we're seeing that play out. Where you have higher HLA-E in the late stage settings where ABC is flatlined, it's always in the presence of high HLA-G and HLA-F.
Do you ever see genetic loss of HLA-E, like deletion
of HLA-E?
Of the gene itself?
Yeah, for sure. Yeah. HLA-E is like smack dab in the middle of HLA-C and HLA-A. So typically you're getting chunks of deletions.
Right.
So,
Thank you.
So John, in the CREST trial, there is a cohort of where they're getting the maintenance is only the checkpoint, and so it'll be interesting data to see if there's improved duration of response. But what my question is that we've got the immunity bio, so you've done a very nice basic science talk, but you've also included clinical. So how do you explain immunity bio where they're giving an NK superagonist when all the cells are bound to HLA and HLA-E and NKG2A? So I don't understand the mechanism of how BCG, you've just said how BCG actually induces all these things. So how is giving more BCG and IL-15 going to eliminate BCG unresponsive high-grade bladder cancer?
It's a fantastic question. For the first time ever in a clinical trial setting, although it was blood cancers, we actually saw a negative result associated with too much IL-15. It's curious. Nobody was ever considering that there could be negative feedback coming from too much prolonged IL-15. I'm thinking about that because we're seeing a very positive response in non-muscle invasive bladder cancer, combining it. I don't have an answer for you.
Okay.
But if it's going to work, we kind of know now that at some level IL-15 is going to have to be self-resolving in terms of signaling to it. It is just like the work done on NK and T cell development in the sort of requirement aspect of IL-15. It never pushed it to the limits. But now that we have these bone marrow transplant studies for AML, ALL showing these negative data, they actually are fleshing it out, and it really seems like there could be too much signaling. So, I mean, we need to look at the data. Right. But my hunch would be that there would be some negative feedback shutting it off, and then probably you're just giving it perhaps unnecessarily.
Okay.
Yeah.
Can I ask a last question? Sorry. So I was just wondering, it's the idea about neoadjuvant BCG. It's kind of sounds a little bit similar to the BCG priming that Dr. Swatik and Joshua Mix has done. And interestingly, as a mechanistic side of it, we have seen in the lab that the gamma delta T cells become cytotoxic and play a role in its efficacy. Have you ever looked at that axis of NKG2A? Because I know the gamma delta T cells also are so I was just wondering what your thoughts on it. And a second question is that same group, they designed this delipidated BCG of, I think, Dr. Torres and Dr. Swatik, that it was initially used in tuberculosis, but it's treated with petroleum, and then it gets rid of the inflammatory lipids. And I was wondering, since you're saying that contributes to this exhaustive phenotype, if that's an agent that holds potential.
Great question. Ultimately, I don't have fantastic response. But we are profiling gamma delta T cells. They're definitely playing a role. We know that gamma delta T cells are not responding to the same antigens as classical T cells. They respond to phospholipid antigens, and they're actually really hard to profile. So there's just no data on like what's driving them. They behave very similar to NK cells, though. They're very sensitive to cytokines compared to canonical alpha beta T cells. But that's, yeah.
Amir, maybe very, very quickly, fantastic talk. I cannot help myself asking here, but I know we're filling the time for sure. But you mentioned with John about some potentially similar data mechanistically in cisplatin resistant, right?
Yeah. We're seeing more than 20-fold increase infiltration of NK cells in the bladder of cisplatin resistant patients, 100% expression of NKG2A, some KIR expression, and we're seeing about like a 4 to 5-fold increase of NKG2A expressing CD8 T cells. And they also co-express KIR, by the way.
That's very interesting. And this is in patients who had cystectomy after neoadjuvant cisplatin-based chemotherapy? Is that the clinical setting?
Yes.
Interesting.
Exactly.
Just makes me think about, and Matt Galsey has said this, whether there are potential differences between cisplatin and carboplatin in the immunogenic potential. So just indirectly, if the mechanisms, of resistance
this is
are
sort
similar.
of the next phase studies that John and I want to get into, is not necessarily comparing carbo versus cisplatin, but just diving into that more clearly. I think this could actually be another indication that follows the same biology.
Great talk, my friend. Fantastic work. Madhuri?
Why not me as well ask a question? No, this is a great conversation, and I'm learning a lot, and I'm almost feeling like shifting from B cells to NK cells. So much excitement. So, how does it differ when there is an excessive immune infiltration prior to treatment versus hot versus cold kind of, or lesser inflammation or infiltration, especially in the cisplatin kind of scenario?
So, sorry, if I understand the question,
In a
you're
cold
asking how does
hot
muscle
tumor.
invasive
Yes,
pre-treatment look?
How
Are you
would
asking?
how would that differ in terms of NK cell infiltration and their positive or negative response?
We're not seeing any discernible pattern that predicts why you would see NK cells in the pre-treated bladder. Some people have them, some people don't. What we can say, and this isn't my work, but it's from Nina Bardwaj's lab, where John is one of the senior authors on it. When you look at untreated disease in bladder cancer and you follow it to later stages of muscle invasion, NK cells are like fundamentally losing their functional sensitivity to cytokines, to doing something about recognizing loss of HLA. Like you can co-culture them with these same lines and they're not functionally responsive. In vitro, when you just let them rest for like 24 or 48 hours, that entire anti-tumor program comes back. And so I'm not sure T-cell biologists can actually say the same thing. So that's a curious finding that needs to be chased down more, but there might be something to exploit there. Yeah.
Potential reversal of exhaustion kind of thing. If you let them rest, for like in the lab, you may see potential resumption of rejuvenation.
Yeah, it doesn't associate with lowering of the inhibitory receptors, though. It just shows them more functionally competent when the inhibitory ligands are not present. So again, we still have to accept, nobody cares about how these cells behave in a dish. It's ultimately in the patient. If the inflammation is going to drive all the inhibitory ligands, then that's a potential problem still. Yeah.
Fantastic talk. Thanks so much, Amir.
Thanks, everyone.
What a great discussion generated. And now we continue on the same pace.
Yes, hopefully. But it was very fascinating, Amir and Neelam as well, a great intro to the whole topic of innate and adaptive immunity. And I'm very excited to be here, actually. And thank you so much, Petros and for hosting this session and making me part of it. So I'm coming from Queen's University, Kingston, where BCG was first given in a bladder cancer patient, and this year we celebrated 50 years of that. It was 1973, and it's very exciting to talk to Dr. Morales every now and then. He says, "What BCG? Move on, do something else. Don't do BCG." He's a fantastic person and a very humble person. All right. So with all the conversation about NK cells, T cells, and macrophages, I think B cells have been kind of ignored in bladder cancer. And probably I think I will do some justice to bringing them into light here. Let's move on. So no disclosures. I'm a basic scientist. And so the B cell and TLS paradox in bladder cancer, I call it B cell and T cell and TLS, tertiary lymphoid structure paradox in bladder cancer, because we are finding two contrasting things when you talk about B cells and TLSs in non-muscle invasive bladder cancer versus muscle invasive bladder cancer. You might have heard or reading the last few years of literature where B cells and TLSs are showing up to be associated with favorable prognosis or favorable outcomes following immune checkpoint blockade or even chemotherapy. But what we are seeing in non-muscle invasive bladder cancer, specifically treated with BCG, these cells and the clusters of these cells are playing a negative role. Maybe playing a negative role, we don't know, but they are definitely associated with a negative outcome. And we do have some answers towards that. And hopefully I can convince you, because it's been pretty challenging when I talk about B cells as negative. People say, no, that's not true. All the data, all these papers show something else. So in this particular plot, as you're seeing on the right side, a lot of these cancers have benefited to some extent from immune checkpoint blockade, such as lung, bladder, melanoma, all these cancers. And a common thing across these cancers is these sites are constantly exposed to environmental antigens and carcinogens and whatnot. Another key feature about these cancers is they have been classified mainly under the inflamed category or more infiltrated category. Or they have more tumor mutational burden, and they have also been shown to have high TLS density and have shown a favorable response to immune checkpoint blockade. So what I feel from a B cell perspective is a fact that has kind of been ignored when we are talking about B cells and tertiary lymphoid structures is the fact that these structures are present across all the mucosal surfaces and all the surfaces that are exposed to the environment. For example, in the nasal tract, nasal-associated lymphoid structures have been quite long known, bronchus-associated, gut-associated, even skin-associated lymphoid structures. And I think gut has been described quite well, and the bronchus. But the bladder tertiary lymphoid structures are just getting attention after we have seen them in all these immune checkpoint blockade, and when we started really appreciating the full tumor immune microenvironment of bladder cancer. So here's an example of how these TLSs, they form as a result of chronic inflammation on bladder mucosa. They do form in acute inflammation as well, but they are kind of self-resolving. But in a chronic inflammation, they tend to stay there, and they mature and they remain energetic. So this is an example of interstitial cystitis, Hunner's lesion that all the urologists commonly see. And this is bladder cancer, and you see something very similar in cystitis cystica as well. So this is a common feature of bladder mucosal immunology, that you'll see formation of tertiary lymphoid structures or lymphoid aggregates or ectopic lymphoid structures that are in the mucosal surface, in the lamina propria mainly, and having the classical features of a germinal center reaction with something like this. So you'll see all different stages of these TLS formation. So with all that background about tertiary lymphoid structures as features of mucosal immune responses and chronic mucosal inflammation, let's now move on to our findings on B cells and what we are seeing in association with BCG response. So initially, we did not start looking at B cells. The reason we went into B cells is because of this first investigation that we carried out in the Euromol cohort. And this was done in 2019-20, around that time at which there were around 460 transcriptomic profiles available for us to analyze. So instead of taking a whole transcriptome-wide profiling analysis, what we did was we had a set of 60 immune regulatory genes of our interest, and we just mined those genes in this entire cohort and divided that cohort into low-grade and high-grade female and male patients. And a specific interest in the sex-associated immune physiology, because immune system is so different in females and males, especially when it comes to mucosal immune responses as well as B cells. So what we found here was majority of these immune regulatory genes were increased or at significantly higher levels in patients with high-grade disease in both females and males. So, and another feature of this particular cohort is females have the shortest progression-free survival. Both females and males did have a shorter, obviously high-grade tumors, they're bad, but females had a significantly shorter when you compared with the other, with the males. So based on that finding, many of those genes were associated with B cell pathways. We designed, and this was work done in 2017-18 when we had smaller multiplex immune panels available. So we had 12 immune markers, mainly T cells, B cells, macrophages. PD-L1 was coming at that time, so everyone puts PD-L1 in their mix, but we also went with that. And what we found was, we found all of these cells, T cells, B cells, FOXP3 cells, tissue-resident memory T cells, everything was higher in patients who did not do well, who had early recurrence. What was most striking was B cells to us, because that was kind of evolving as a marker of positive response in many other cancers. So we focused on B cells, and we had some data on macrophages, M2-like macrophages as well. So what you're seeing is increased number of stromal B cells. Mostly they're closely associated. The correlation between M2 macrophages and B cells was significantly higher. We use the pan B cell marker, CD79A. B cell has many subsets like any other immune cells. They come in whole different flavors and populations. So we just use the pan B cell marker. And what we are seeing is increased numbers of these. When tumors have increased numbers of these, the patients have a shorter recurrence-free survival. And again, even in our local cohort of 332 patients that was assembled by our pathologist, Dr. David Berman, what we see is females have the shortest progression-free survival here, and which has been kind of variable in different cohorts, but here these two cohorts definitely show that kind of profile. So this cohort of 332 patients some of them were treated with BCG and some did not undergo BCG treatment. So, and B cell population, increased number of B cells did associate with everything, irrespective of the treatment, in terms of shorter recurrence-free and progression-free survival interval both. So we just took the tumors from patients who were treated with BCG, and what we find is the exact same finding: increased number of B cells associated with shorter recurrence-free survival and progression-free survival as well. And again, females had the highest and the shortest survival. And the numbers of B cells in females, stromal B cells, were significantly higher. This particular paper was just recently published. I don't know how many of you have seen it. It's a Science Translational Medicine paper that was recently published by another group. And whenever we have this, because for non-muscle invasive bladder cancer, there are not too many cohorts out there where we can profile and look for genes of our interest. So we looked at the CD79A profiles in this particular cohort as well. While we see that both females and males, increased expression of this particular gene is associated with poor response, but the sex difference is kind of reversed in this particular cohort. So this is kind of interesting, but overall, this again confirms that B cells are associated with bad outcomes when treated with BCG specifically. So where are all these B cells located? We generally don't see them. I think one major reason because B cells were not encountered before because they are not heavily infiltrating in the tumor epithelial compartment. So we went back and looked at the whole tumor sections of these patients, and what we saw was most of these B cells, our first work was done on a tissue microarray. We can't stain 332 whole sections with multiplex IF, so there was a TMA constructed with a core size of 1 millimeter. So this is how they looked. And when you go back and look at the whole sections, you can appreciate that the TMA sometimes does not capture the stromal environment really well. It may or may not. There can be errors, and which adds to some of the errors that come up in our analysis. So we went back and looked at the whole tumor sections, and what we saw was these lymphoid aggregates or tertiary lymphoid structures were very closely associated with the tumor epithelial compartment in patients who did not do well or did not respond well to BCG treatment. So we stained them with multiplex immunofluorescence, markers that characterize tertiary lymphoid structures, and what we found was different stages of these TLS formation from as it occurs in a classical germinal center reaction. You'll see something very similar early to fully mature tertiary lymphoid structure that has high endothelial venules and other features associated in this closed network of follicular dendritic cells. So you encounter all these kind of scenarios. But what was very interesting was and it was present in both BCG responders and non-responders. And what was very unique or very interesting was patients who did not do well, the BCG non-responders who had recurrence within 1 year of their initial TURBT. The fully mature TLSs with presence of PNAD positive high endothelial venules were higher in patients who did not respond to BCG immunotherapy or recurred earlier. So the next question was, what do these TLSs consist of? What do they express? Because we are seeing them both in responders and non-responders, albeit the difference in the numbers and density of those. So we use the nanostring GeoMX digital spatial profiling technology to specifically characterize the epithelial compartment and the TLSs and look for 40 immune-related proteins. These were scattered across 3 different panels. And what we saw was, irrespective of them being in the non-responders or responders, these were highly expressed, all the immune checkpoint related or immune exhaustion related proteins, and more immune cells were proteins that reflected of the immune cell abundance, including NK cells, of course, were present in the TLSs, and mostly exhaustion related proteins, as I said. There was no difference between the levels of, or there was no significant, statistically significant, I should say, between the TLSs from non-responders and responders, because that reflects the chronic inflammatory state. So the next question with all that patient data comes, how do B cells actually respond to BCG, and why are we seeing this very contrasting and counterintuitive finding that high B cells and increased TLSs associate with poor response to BCG? And is it practical at all to target these exhausted populations of B cells for improving response to BCG? And the picture you're seeing here is just a mouse bladder we had put in BCG, and we just stained with the Zeal-Nielsen staining that is used in microbiology lab just to see how BCG enters. So this was immediately within 2 or 3 hours of just installation in the mouse bladder. It's just a curiosity question. How does it traffic into the bladder microenvironment? So when we look at the factors that are associated with BCG unresponsive state, there are several tumor intrinsic and host intrinsic factors, and this has been studied quite widely ever since BCG came into existence in bladder cancer treatment. So the tumor stage, obviously, the tumor stage and tumor intrinsic factors, but factors such as the age and patient sex or gender have not been quite well studied. And given the mucosal inflammation and given the differences between female and male immune system, we thought of considering these 2 factors in our mechanistic and in vivo modeling studies. So Just to re-emphasize, this is data that is not our data. This was very recently published in 2020, where they are showing that in females and male, in normal healthy individuals, you do see, as we age, because of immunological aging, immune senescence, there are significant shifts in the immune cell populations, and with aging, a population that significantly differs in females and males is the B cell populations. Females have more of B cells in their circulation, despite the depletion in periphery and despite them trafficking to many mucosal sites. Females have more compared to males. And another key feature is, another important finding is chronic exposure to all these, let's say CMV, age, smoking, sex. There are significant age-related shifts that are seen in the peripheral immune cell populations. And as you're seeing here, it's mainly age, smoking, and sex. All these affect B cell populations, and CMV is mainly affecting the T cell population. Of course, CMV affects all different immune cell populations. But this was kind of interesting, and this made us think about how we should be actually modeling to study B cell responses to BCG. So what we did was our first investigation before we initially started our work in mouse models of bladder cancer. We first tried to incorporate these factors, study how a healthy bladder ages and what changes as the mouse bladder ages. So this study we published a couple of years ago, where we characterized the baseline features of mouse bladder with respect to the sex and age of the mouse. And what we found was very interesting, actually. As you're seeing here, as the mouse ages, so the age groups we included was from young to old, 3 months being sort of a young adult mouse, and 18 months being kind of a 60, 70-year-old human. Mice are not human, we all understand that, but they very closely reflect the immune system changes that happen on our mucosal immune system, or even peripheral immune system. Systemic immune system. So what we saw was, as the mice age, their immune cell landscape changes in the bladder. So we just took healthy bladders from male and female mice and subjected them to RNA sequencing. And what we are finding is, around 9 to 12 months of age, there's significant shifts in the B cell associated pathways. And this gets enriched more in females compared to males. Both male and female experience the same, except for the fact that females experience this early, given the fact that many of the immune-related genes are located on the X chromosome, and many of them escape the X chromosome inactivation. Now, the X chromosome inactivation phenomenon has not been really well studied in mice, and we know that the number of genes that are escaping is much lesser than what happens in humans. But one thing that is very clear, that with age, this process of X chromosome inactivation is not very efficient. So there is leakiness with aging. And what we saw was, with aging in healthy bladders, TLS formation was more evident in female mice. And this has been shown by another group as well, where mice were raised in germ-free conditions, and you still see the same situation with the same kind of scenario, with, in a non-antigen dependent fashion, TLS formation as a result of increased TNF-alpha occurs on mucosal surfaces, such as bladder as well. So, with all this, now the next question was, why are these B cells behaving bad, and what happens with aging to B cells? So we came across this population of B cells called atypical B cells. These were described as age-associated B cells a while ago, almost 15 years ago. And they have been described as a subset of memory B cells, where when exposed to interferon gamma, IL-21, TLR7 or 9, they acquire the CD11C and FcRL5 and increase their T-bet expression. And they kind of behave in an exhausted or energetic manner. And with aging in mice specifically, or in chronic conditions or autoimmune conditions, or booster vaccination, an increase in this population of cells has been shown in humans as well. Again, this was more interesting to us from the mucosal immune perspective, environment perspective, because of interferon gamma being abundant in the bladder microenvironment after BCG goes in, and TLR7 more because of the cell death that's happening because of tumor cell death, and TLR9 because BCG does go through TLR9 pathway as well. So, which is why we focused on this particular population of B cells. So the first thing, again, what we did was we really wanted to confirm that we are using the right age group and right, and profiled, and the kind of incorporating the sex aspect as well. So we took healthy aging female and male mice, 12 months age, which is kind of reflective of a middle-aged human. And we just characterized their peripheral atypical B cell population, which is characterized by decrease in CD21 expression, which is a memory cell marker, and increase in CD11C expression. So what you're seeing here is total B cell number did not change as much, whereas atypical B cells were significantly higher in female mice compared to male mice. And even in male mice, this happens, the increase happens, but at a later stage of their age. So we then moved on to the BBN carcinogen-induced mouse model, because it's a nice model to study disease progression, and our question was related to disease progression. And as we all appreciate, there is no perfect mouse model for bladder cancer, but I think this is a very cool model that the bladder we all have, because we can really study chronic inflammation and mucosal inflammation associated with chronic carcinogen exposure, such as cigarette smoke-induced, mimicking the cigarette smoke-induced carcinogenesis process. So what we did was we ran the study in both young and old mice, but most of the data, we moved on to study in older mice, because that's where we were seeing something interesting related to the atypical B cell population. And this is kind of a schematic of what we did. Both female and male mice old mice were exposed to BBN through water, and B cell depletion was initiated right before BCG was started, around 7 weeks of age. So in aging mice, you'll see tumor incidence happening earlier compared to younger mice, where 12 to 13 weeks to 15 weeks has been shown as the time point for treatment initiation. But if you use aging mice, you'll be able to see that those events happening early because of immune exhaustion and immune senescence related features. So we depleted using a cocktail of B cell targeting antibodies, and then treated these mice with 3 weekly doses of BCG, and maintained the depletion in one arm, in both female and male mice. So this shows that TLSs do form because of BBN exposure in more predominantly in female mice compared to male mice. And what you're seeing is, this is a healthy bladder here, and after week 7 post-BBN exposure, you have these TLSs forming. After the first dose of BCG in female mice, you do see this mucosal healing happening, urothelium returning to its benign to normal states. And after the third dose of BCG, around week 6 to week 8 post initiation of BCG treatment, we see the disease progressing again despite removal of BBN carcinogen. So there is something going on here because of excessive inflammation. Maybe some residual cancer cells are progressing. So that kind of indicates the negative role of these B cells. What we saw was in the depleted females, so this is on the left, you're seeing in the non-depleted females and males, there is more B cells in the bladder microenvironment in the lamina propria compared to the male mice. And when you deplete, there are still a few residual populations of B cells still present in here in the female bladders, whereas in the male bladders, you're seeing that they're almost completely depleted. So that was the first confirmation we had. Another very important finding from this study, and very interesting finding, was B cell depletion during BCG treatment. It augmented mucosal healing. So the urothelium returned to its benign to normal state despite the presence of, so we had 2 arms, BBN with BBN and without BBN, and in both the scenarios we see that the mucosal healing is more pronounced in female mice when you remove B cells during BCG treatment compared to the non-BCG treated non-depleted mice. And TLS formation was again minimal in these mice when B cells were depleted. So here you're seeing an example of post-BCG. You have, I'm sure many of you know granuloma formation is a classic feature of BCG infection. So even in patients, it has been shown quite variably that granuloma formation happens after BCG treatment, which is what you're seeing here. And because of excessive interferon gamma in the microenvironment, you're also seeing PD-L1 increased with all the macrophages, a suppressive macrophage population. But when you deplete B cells during BCG treatment, you'll see the PD-L1 expression has significantly reduced, along with the mucosal bladder lining coming back to its benign to normal states. We also measured the plasma cytokines from depleted male and female mice that were subjected to BCG treatment, and what we found was very interesting. Both Th1 and Th2 cytokine levels were significantly increased in female mice that underwent B-cell depletion during BCG treatment. For example, IL-2, interferon gamma, GM-CSF, Th1 cytokines, and IL-9, IL-13 as the Th2 cytokines. So that was kind of a favorable outcome. We also profiled the plasma immunoglobulins, and we see the memory B cell population increasing because IgG2b is mainly secreted after class switching in the memory B cell population. So we see that happening as a result of BBN exposure, as well as a result of BCG treatment, and as a result of increased lymphopoiesis in female mice, because B-cell depletion does initiate lymphopoiesis. So we are seeing that effect very clearly. When we look at the bladders, and we conducted the gene expression profiling of bladders collected from these mice, and what we see is in the depleted mice, there is increase in expression of CXCL10, TNF-alpha, decreased IL-17A. So the kind of a favorable kind of immune environment generated because of depletion of B cells. We've also subjected B cells isolated from male and female mice to be exposure to BCG as well as TLR7, because TLR7 initiates makes B cells differentiate into ABCs and increase their survival. So I'm not showing the TLR7 data here, but what we found was when BCG enters, when B cells are cultured in the presence of BCG or IL-21 and interferon gamma that are required for B cell differentiation into ABCs, you see an increased expression of the immunosuppressive IL-10, and that is again more dominant in the female B cells compared to the male B cells. So these were pure B cell isolated from the spleens of mice that were exposed to BBN carcinogen. And in green is CD11C, which is a marker of atypical B cell, and in red is lipoarabinomannan, which is staining for BCG entering in the B cells. Now, the question of sex differences, why are we seeing this huge difference in male and female B cells as well as response? We moved on to the 4-core genotype mouse model, where we can nicely study the sex chromosome and hormone-related effects. And these are, again, aging mice, where we subjected them to BBN exposure first, and you see clear differences in their tumor volume as the disease progresses, males having a higher tumor volume irrespective of the sex chromosome complement. In these mice, again, you're seeing that when there is 2 X chromosomes present, there is more TLS formation happening in the microenvironment compared to all other genotypes, whereas in male mice that get additional X chromosome, you see the TLS formation initiating as well. So there's something going on with the X chromosome associated genes here, probably TLR7, which is located on the X chromosome, which mainly escapes X chromosome inactivation. When we characterize the bladders using RNA-seq, if you just follow the red bars, you'll see all these immune-related genes, including the atypical B-cell marker genes, are higher in female mice that has the X chromosome, double X chromosome complement compared to all the other genotypes. And when we treat these mice with BCG, we again see the same kind of profile, female mice having more atypical B cells, and when they are treated with BCG, you're seeing that the numbers are significantly going down, but this magnitude difference makes more B cells traffic to the bladder microenvironment. And in the bone marrow, you just see the opposite shifts when there is an increase in the atypical B cells in the bone marrow. So what does all this mean? Just to summarize very briefly, I think in a BCG non-responsive scenario, what we think might be happening is there is a chronic carcinogen exposure. There's basically an immune exhaustion that is happening because of a chronic carcinogen exposure, because of age-related immune exhaustion, because of carcinogen exposure-induced immune exhaustion. And that happens in a sex differential manner. Females have more B cells, so you see more of atypical B cells accumulating in the systemic circulation of females compared to males. So in patients, but the fact that B cells and TLSs are associated with bad outcomes following BCG in both the sexes, it's possible that in patients who have more TLSs and B cells, it's kind of reflective of all these events. And when this patient gets more repeated doses of BCG, it's pretty much exactly what Amir had just described in the context of T cells. We are seeing exhausted populations of B cells, which are very critical to mucosal immune response, expanding, and when they are trafficking to the bladder, you see a suppressed microenvironment. And we are seeing something similar in the patient response as well. This is a very small set of patients that we have characterized so far, and this study is still ongoing. But what we are seeing is, in patients, it's a very small set, as I said. We have so far done 25 patients, and what we are seeing is after the 4th BCG installation, this population of B cells, the atypical B cells, increases in patients who experienced early recurrence. So that's kind of reflective of this whole phenomenon that is happening. So with that, I would like to end my talk. Thank you again, everyone, for listening. All these people who have been instrumental, Priyanka especially, and Gonel, they have done most of the work, including all these people here. And all the clinical support that I get is from Dr. Rob Siemens and our pathologist, Dr. David Berman, and all the other clinicians, all the other urologists. I don't think I have left anyone in Canada that I have not approached for questions. Alex is sitting right here. And the funding for this particular project was provided by all these agents. We really thank the patients who consent for our studies and the animal care facility at Queen's. And thank you to BCAN for having me here and present our work.
Thank you, Madhuri. Fantastic work, very elegant and very, very thought-provoking. We're just thinking here with Neelam, maybe we can have Keith give his talk, and then maybe we can do questions at the end. Is that okay?
Absolutely.
Perfect, perfect. Okay. So maybe you can upload Dr. Chan's talk via Zoom. Keith couldn't come today, unfortunately, but he's with us virtually. Thanks for uploading the. Yeah. Eventually will. Yeah. I think it's a pre-recorded talk, if I remember correctly.
Should I? Petros? Should I get started, or?
Keith, can you hear us?
Petros and Neelam?
Yeah, we can hear you. We can hear you. Thanks for joining. I think it was a pre-recorded talk, right? Yeah. Rebecca is like he's live. I can hear him. Yeah, I think his talk is recorded, but he joined for the Q&A. I think that was a plan. Yeah. Maybe we can take a question for Madhuri in the meantime.
Sure.
Yeah.
Do you want me to go to the podium, or I can answer from here?
Oh, from here is fine. Yeah. Alex, you have a question, I can tell. I can feel. Thank you.
I mean, the classical teaching is that when you have tuberculosis and people were only building a humoral response, no cellular, they were actually dying. And now there's the interplay. But the question I have looking at, I was thinking, so several groups ourselves 25 years ago showed that basically if you have a humoral response, and we dig into it, it's actually a poor prognostic factor, fine. But in tuberculosis, where if you assume that women have a more pronounced B cell component, and there are so many patients who are affected by tuberculosis, do we have any data that actually once they actually, women have an imbalance, or those who drive more B cell, anything of this kind that to support this?
Yeah, I think in the context of immune response to BCG vaccine, I think girls respond better compared to boys. That is known. In terms of B cell response, the follicles that form in the bronchus are larger in women or even female mice compared to male mice. That has been also shown. Now, B cells have not been really studied quite well in tuberculosis, but there is one recent paper that came out last year, or this year, earlier this year probably, that talks about B cells and a little bit about these atypical or tissue resident kind of B cell populations as well. So they are evolving and they are appreciating the role of humoral immune response a little bit, but there's not a lot of data available. But it's a classic mucosal immune response, granuloma formation, and granuloma is full of B cells, mainly with some macrophages.
I'll let Amir eventually jump in. The second question was about, I mean, the antigen receptor has been suspected to have some interplay, although it's very debated. So do we have any idea about how that interplays with the B cell response, generally speaking, and then in particular when submitted to repeated stimulation?
Sorry, can you repeat the question, Alex?
The antigen receptor has been suspected.
B cell antigen receptor.
And antigen receptor in general, correct, on some of the cells. But what's the interplay? And I don't think, to the best of my knowledge, I don't think there's a good understanding between the antigen receptor and actually the B cell function.
Yeah. Actually, atypical B cells specifically, they get stimulated via non-B cell receptor pathways. So the complement receptor, CR2, that is present on B cells, those take up, the B cell, BCG antigens. And there is one study that came out, I guess 1993, it's an old study, that's the only study that has shows CR2, which is CD21, and BCG response, or taking BCG in by B cells because of that particular receptor. So it's not really widely studied, but that's how, it's not just the B cell receptor. There's a non-B cell receptor pathway that is acting in these cells.
Very interesting. You have a comment, Amir?
Yeah. So the B cell function itself, it's very curious, and you're seeing that it's obviously affected by chronic activation as well. We found that where interferon gamma is like the single most dominant signal that we saw in the resistant specimens, that IgG1 and IgG3 and plasma cell markers were the second most strongly upregulated pathway that we saw. So you're asking your question about the antigen specificity. It's obviously, it's a great one. But what about the FC fragment of those antibodies? So in humans, it's exquisitely specific. It's about IgG1 and 3. And in your mice, it's going to be IgG2A and 2B. Have you looked at these TLSs and...
So we haven't stained because, but I think there is a signal for, we have looked at these signatures in tumor profiles, and we see all these in patients who did not respond to BCG. So it's there. I think it's also an ADCC kind of phenomena that's happening that we haven't looked at all. But yes, if you stain these for IgG or immune complexes, I think we will find them there.
So it's curious because it means that B cells are like promoting a process that's ultimately going to result in their becoming these atypical phenotypes because the gamma is driving.
Yeah, it could be both ways, that are recruited versus that are locally present because of chronic inflammation. Yeah.
Interesting, fun, interesting, very elegant biology. Thanks so much, and thanks, Alex, for the question. I think, Keith, you can hear us now, so we'll let you give the presentation. Thank you so much for joining us. Keith, can you hear us? I was too optimistic, I think.
So Petros, I can only hear Neelam and Madhuri's microphone. I cannot hear yours.
Keith, can you hear us now?
Yes, I can now hear you.
Awesome, awesome. Thank you so much. We'll let you go ahead with the presentation. Thank you so much for joining.
Okay, thank you. Thank you, BCAN, for the invitation. And Neelam and Petros put together such a great sessions. I'm definitely missing out here, meeting with colleagues and missing out many good discussion. So due to the time, I'm gonna cut to the chase. I hope to show you a story that we, in the muscle invasive setting, which is a good segue from the non-muscle invasive data, that we hope to convince you that IDAM blockade is a innovative approach to enhance chemoimmunotherapy response in the muscle invasive bladder cancer setting. So I borrow a slide from Neelam from her outstanding overview, showing the highlight of our story. As you know, that in the immune checkpoint blockade setting, most people are focusing on T cells, and we show here we show that in our previous study shows that dendritic cells is crucial for priming a antigen-specific T cell response to enhance immune checkpoint blockade therapy. So, as you know, this audience already well aware of the importance and revolutionized of immune checkpoint blockade therapy in the advanced bladder cancer setting. Since there are trainees and patient advocates in the room, I just want to briefly highlight that T cell activation requires MHC1 activating T cell receptor for signal 1, activating signal 1, as well as CD80 or 86 to activate a co-stimulatory signal, so-called signal 2. And as Amir earlier mentioned, there's also a signal 3 by cytokine activation. And in cancer setting, due to chronic antigen exposure or oncogenic signal, cancer cell as well as tumor or myeloid cell can overexpress PD-L1, which binds to PD-1 on T cells that blocks signal 1. And T cell, when overactivated, can also overexpress CTLA-4, which competes away the co-stimulatory molecule to block signal 2. So in the bladder cancer field, we investigated has been trying to combine immune checkpoint blockade therapy to enhance the patient response with chemotherapy. Hopefully that 1 + 1 = 2. However, 2 major phase 3 clinical trials combining chemotherapy with immune checkpoint blockade therapy do not result in very desirable clinical outcome. This is one led by my senior colleague, Mukherjee, showing that combination therapy did not increase objective response over chemotherapy. Therefore, this is a very significant clinical issue. So, but from the retrospect, it might be slightly counterintuitive to combine chemotherapy that are historically known to be immunosuppressive or myeloid suppressive. Although there are hopes that chemotherapy can release new antigens into the to enhance an immune response. When we survey the literature out there, in fact, there are certain chemotherapeutic agents known to induce so-called immunogenic cell death. And majority of them are not standard of care for bladder cancer treatment. And cisplatin or gemcitabine are not among the chemotherapy known to induce immunogenic cell death. So to this audience, maybe not everyone familiar with the mechanism of immunogenic cell death activation. So this is work done by my former student, Kazu. So-called immunogenic cell death depends on translocation of keraticolin that from the ER to the cell surface, so-called eat me signal that recruit antigen presenting cells such as dendritic cells, as well as release of damage-associated molecular patterns or DAMs, or DAMs danger signals such as HSP70, 90, or HNGP1 that engage pattern recognition receptors on dendritic cells, rendering mature and to activate and prime a effective T cell response. And this is data from our lab showing that gemcitabine, although not thought to be a immunogenic cell death inducing agent, our mass spec analysis shows that they can also induce the release of cell surface activation of these classical DAMs. So at the time we were puzzled because, and this phenomenon can be reproduced in both multiple cell lines as well as in the pancreatic setting. So at the time we were slightly confused because a non-immunogenic agent, gemcitabine, able to induce these classical DAMs that are known to induce ICD or immunogenic cell death. So what we did is we first validate that these findings are valid by flow cytometry, showing you can see CRT in the cell surface, HSP70, as well as release of HMGB1 into the supernatant. And then we turn to a classical so-called vaccination assay to evaluate whether a drug is ICD or not. So this assay is straightforward. So we treat IC50 dose of a drug, induce 50% of cell death, and use it as a vaccine to inject into the left side of the mice. So if this is a ICD-inducing agent, 7 days after when we inject the live tumor cells, it will reject the tumor. And this will be the tumor response percentage of tumor-free mice in an ICD-inducing agent will look like. Contrastly, in converse, if it is a non-ICD-inducing agent, you won't be able to mount a tumor-specific response, and tumor will form, and you will gradually see the more and more tumor-free, less and less tumor-free mice. So, as expected, a positive control chemotherapeutic drug, mitoxantrone, is shown to be an ICD-inducing agent. The negative control cisplatin is known to be a non-ICD inducer, and we found that gemcitabine, surprisingly, when they were able to reduce the release of DAMs into the supernatant or in the cell surface, they remain a non-functional ICD-inducing agent. So then we turn to another pathway, the COX-2 PGE2 pathway that was previously shown by us to be a mitogen that is released by dying cancer cells to induce repopulation of residual cancer stem cells. And we found that in this G69 syngentic model that we found in the mouse, that this COX-2 and PGE2 pathway is also induced. So we hypothesize that in additional to the dogma where we think the investigator shows that DAMs are the predominant determinants of immunogenicity versus tolerogenicity, there is a inhibitory DAM pathway, and therefore is a balance of these DAM and IDAM, inhibitory DAM, in the tumor microenvironment that determine tolerogenicity or immunogenicity. So we use a drug and that can block COX-2, and most recently we also use a downstream drug that blocks the receptor for PGE2, the EP2 and 4 antagonist, as a functional drug to block this pathway. So what we found is we can convert by blocking this pathway using drugs or a genetic background. We can convert a non-immunogenic drug, gemcitabine, into a immunogenic drug. And we also found that this is T cell dependent. When we deplete CD8 positive T cells, this phenomenon is being abrogated. And I just want to show you that this is dendritic cell dependent. Due to the time, I just show you that this is we adapted a footpad assay where we can detect these dendritic cells in the draining lymph node. We repeated that assay, and we show that there are more CD103 positive dendritic cells that are drained into the lymph node. And we found that the signal 1, 2, and 3 that I mentioned earlier are being enhanced or induced in these dendritic cells, indicative of their activation status, as well as a lymph node trafficking signal. And we also show that in the T cells, there is higher, TC1. In the TC1 or polarization is a marker indicative of TC1 is being induced, and the markers for Tregs are being repressed in when we block this IDAM pathway. And there are the T cells are being activated as well. So to this audience, you want to see this in a more preclinical setting that make the clinical setting, right? So I don't need to convince you that they're inflamed or immune excluded or cold tumor. However, the current model in the field, such as MB49s, are the mostly inflamed tumors that are more favorable to the immune checkpoint blockade therapy. And more than 50% of the patients are immune excluded. So here we show that that we have developed a syngeneic model called G69, where you can see the T cells marked in green here are either excluded at the tumor periphery or being stuck into the stromal that is marked by these alpha SMA positive stromal cells. So our hope is to block IDAM to enhance so-called signal 0, and therefore enhance tumor DC maturation, activation, and priming. So here you can see this G69 model recapitulate the clinical setting where you can see the vehicle, GC treatment alone, GC plus PD-1 alone, as well as GC plus PD-1 are stacked up here. They're highly resistant. And when we combine with IDAM blockade in the presence of PD-1 or in the presence of PD-1, you can see that combination therapy significantly enhances tumor response. And we have data showing that we are not showing here is that they're rendering DC activation, as well as you can see these functional T cells, the frequency is being increased when there's IDAM blockade. And this is work done by Fotis, a talented assistant professor in the lab. So then Fotis recruit other students, Zan and Mark, to look at how this IDAM blockade strategy can we flip like cold tumors into inflamed tumor. You can see here that when we treat combination with IDAM blockade, you can see more T cells are being infiltrated into the epithelial region. And when we use the CD8 positive antibody to deplete, we show that these T cells are functional, while a depletion strategy in the GC only setting does not work. So this is maybe a more clear to the audience, showing you that combination of IDAM blockade brings the T cells from the periphery into the epithelial region. And this is clinically relevant, this pathway, since we also look at MVSc 210, where we have pre-treatment tissues showing that PTGS2, the COX2 pathway, is upregulated in the non-responder. And high expression of PTGS2 or COX2 PG2 pathway associate with a poor response in this ICI MVSc setting. And just want to mention that this is not only a bladder-specific phenomenon. In a group published similar data in UK in the breast cancer setting, that combination treatment of celecoxib also enhance tumor response as well as control metastatic lung foci. So I hope to convince you that we are now moving on to a IDAM blockade strategy to try to flip the TME into an immunogenic setting that can combine with ICI and chemoimmunotherapy strategy. So we are now, as I mentioned earlier, we are trying to obtain these EP2 and EP4 dual antagonists to combine with chemoimmunotherapy. And we are trying to generalize this to other drugs, such as there's a lot of attention about radiation. And my colleague, I just want to give a shout out to my clinical colleague in Methodist, Dr. Raj Sakuna Sivan, who is running a radiation plus ICI. It's a phase 2 trial, single arm with 33 patients, combining radiation with ICI. And you can see here, radiation really suppressed this IDAM pathway. Maybe one of the strategies, one of the mechanisms why radiation is having a good response. This trial is now with 42% accrual and very remarkable safety, as well as PT0 downstaging efficacy. And here you can see that there's another drug, cabozantinib, that is getting a lot of attention in inducing a T-cell response. But you can see there is a COX-2 significant enhancement, suggesting maybe combination with the IDAM blockade could enhance the immunomodulatory effect. So I just want to thank my team. I thank them throughout the funding source, especially the this is a patient fund. Everything that started from a patient funded study where unfortunately passed away from a metastatic cancer, and also want to highlight the trainee funds, and we are rapidly expanding our research program, translational research program at Methodist. Just want to thank you for your attention, and I cut it within the time. Thank you very much.
Thank you so much, Keith, for a fantastic talk. Very interesting, and the COX-2 story keeps evolving over time. It's interesting to see that. I think we're actually on time, but maybe one quick question from the audience, or from the panel, for Keith. Is there anything in the audience? Amir, any comments?
Yeah, Keith, can you hear me?
Keith, can you hear us now?
Yes, I can hear you.
Fantastic, thank you. We tried to find the right microphone here.
Sorry about that.
Fantastic talk, and thank you so much for connecting, and really elegant work with an evolving, story about COX-2, and a very interesting trial design with the sasanlimab and the radiation. That's an interesting concept. Looking forward to see the results. If we have time, maybe for one question, and I think Amir next to me has one.
Yeah, Keith, thanks a lot. A really beautiful talk. I wanted to ask your insights, given what TGF-beta is doing to the COX-2 pathway, and given what we're seeing with it inducing this sort of divergence from these PD-1-defined CD28 negative T cells. What's your thoughts about sort of what I presented on that biology? It's not in mice what I showed, but it's sort of looking at similar stages of disease. What do you think COX-2 is doing in this?
Yeah, so I mean, great question. I hear bits and pieces in and out, but I think you asked about the TGF-beta or the NKG2, your own pathway in this setting, I believe.
Yeah, but, sorry. I was just asking insights onto if you think those pathways are interacting. That's probably an easier way of phrasing it.
Well, definitely, right? So as we previously want to pursue chemotherapy setting, we also see the subset of NKG2 your NKG2A being upregulated. And we mentioned there is like a HPK, which can be the upstream of PGE2, which we hope to be trying to pursue in this setting. But I think there are many opportunities in this chemotherapy treatment, in the combination treatment in the cisplatin ineligible setting, as well as in the neoadjuvant setting, for combining these type of IDAM blockade strategies or strategies that can induce immunogenic cell death. And also I missed to mention that the ratio of these DAM and IDAM in the serum as well as in the urine could potentially be used as a selection marker to predict responder versus non-responder, right? Which we hope to move forward in this setting. So I hope I partially answer Amir's question. Yeah.
Thank you so much, Keith. Can you hear me okay now? No.
Yep.
Okay, I think he hears us, right? All three all together.
I think it's Amir's microphone.
That's the way to do it. Keith, thank you so much. This is really, really interesting. Maybe before we go, very quickly, can you comment a little bit on the trial design that you presented with a anti-PD-1 mechanism with radiation, and what's the readout, and how many patients you plan to enroll? The sasanlimab radiation.
Yeah, so that trial is correct. That trial is by my clinical colleague, Dr. Raj, and he's enrolling 33 patients that are in the cisplatin ineligible muscle invasive bladder. You can see this clinical trial number here if you would like to get more information. I'm trying to get him more exposure. This is trial driven by him, and we think there are intersect. So this is now 42% accrual. So he's very conservative, but we are seeing great PT0 response. And obviously, with me here, we hope to obtain these patient tissues, also look at, as well as urine and blood, to test our concept where the IDAM and DAM ratio could be indicative of early response to this combination strategy, and which can be hopefully extrapolate to other ICI setting. So I would hope that P can get attention, and it can invite Raj into future think tank activities. And I think he would be a very good addition to the audience.
Very, very interesting. Keith, thank you so much for joining. Fantastic session. You know, we actually were able to fill up the time, and we think of Rob, of course, Dr. Svetak, and thank you so much, all the panelists. Neelam, thank you for being a fantastic chair. Thank you to all of you. Thanks for the audience too.
Yeah, thank you everyone, and thank you for the chairs, putting together a very enjoyable session. Thank you.